Hello, this is video number 2 for Module 5, and we're discussing extensions from Mendelian inheritance. Basically, things that Mendel didn't see and genetic phenomena that extend. They don't disprove or counteract. They still are within the framework of Mendelian inheritance, but they extend what Mendel saw. So here are the three main topics we'll cover in this video. Genes with multiple alleles. By multiple, I'm meaning more than two. Mendel saw only examples of genes with two alleles. P color, allele for yellow, allele for green, P shape, an allele for round or an allele for wrinkled. We're going to look at examples for genes that have three alleles or more than three, or even an example kind of an extreme example. I'm going to be talking about three of these examples. The PowerPoint will have information about all four. Second thing we'll talk about is pleiotropy, and pleiotropy is a gene that controls more than one trait. So mutation of that gene will have more than one effect on the phenotype. And finally, we'll talk about alleles that are lethal in homozygous form. So they're called recessive lethal alleles. So starting with the first topic, genes with more than two alleles. So this is actually the norm. If you think about a gene as a stretch of DNA, any of the nucleotides in the DNA can be mutated. And so really the number of alleles is dependent on the number of different nucleotides. So in the examples that we'll talk about, the first one is the ABO blood type antigens. And we talked about this last time as an example of codominance, and we're going to add on to that. Secondly, an example of genes controlling the seed spotting pattern in seeds of the lentil plants. And the third example is antigens that occur on human cells called the human histocompatibility antigens. All right, first example, the ABO subtypes that are determined by alleles at the I gene. And last time, the first video, we talked about the A allele and the B allele. These alleles control production of a glycoprotein, and the sugar is the thing that differs between those two types. So the A allele controls synthesis of one type of sugar, the A type. The B allele controls synthesis of a slightly different type. Now there is a third allele at this gene, and that's the little I allele. And little I is non-functional. It's a null allele. So it produces neither the A type nor the B type. Now, in different genotypes containing combinations of two of any of these three, these are the phenotypes that we'll see: genotype, phenotype. So if you're homozygous for the A allele, you produce the A type sugar. However, if you're heterozygous for A and little I, you also produce only the A type sugar. If you're homozygous for the B allele, you produce the B sugar, and similarly to A, if you're heterozygous for the B and the little I, you produce only the B sugar. If you're heterozygous for the A and the B allele, you produce both A and B sugars. And that's the codominance example from the previous video. If you're homozygous for the null allele, little I, you produce neither A nor B. And so that's blood type O, absence of both A and B antigens. Notice that there are two types of dominance relationships between alleles. There's codominance, seen for this heterozygote, producing both A and B. And there's complete dominance, seen in these heterozygotes, B with little I and A with little I. In that case, only A is produced or B is produced. So codominance and complete dominance. Okay. Second example is the seed coat pattern, meaning what is the pattern of pigment seen on the surface of seeds from lentil plants. There are 5 different alleles of a gene called the C gene, and we talked about 2 of these already, the spotted and the dotted. In addition to those, here's this phenotype of spotted big blotches and dotted small dots. There is an allele for clear seeds, basically no spotting pattern at all, no blotchy pigment. And then there are a couple of other alleles called marbled 1 and marbled 2. Here's the marbled 1 phenotype. Here's the marbled 2 phenotype. They're similar, but there's difference in intensity and the pattern of marbling. Okay, so there are 5 different alleles. And if you make heterozygotes for all combinations of these alleles, then you can define what's called a dominant series. For example, if you make a heterozygote with marbled 1 and any of the 4 alleles, you see the marbled 1 phenotype. That means marbled 1 is dominant to all of the others. If you make a heterozygote with a marbled 2 allele, and 3 of the other alleles, not marbled 1, but the other 3, marbled 2 phenotype is seen, indicating that the marbled 2 allele is dominant to the spotted, the dotted, and the clear allele. If you make a heterozygote with the spotted and the dotted alleles, this is what we talked about last time, you see codominance. You see both the spotted and the dotted phenotype produced. So the equal sign here is indicating these are codominant. And finally, if you make a heterozygote with a clear allele in any of the 4 alleles, the clear allele is recessive to all 4. So that's what a dominant series is, indicating which alleles are dominant and recessive with respect to the others. Final example of genes with multiple alleles is an very extreme example. And this comes from the genes encoding human histocompatibility antigens. These antigens are found on the surface of all body cells in humans except for the red blood cells. And they're important for the immune system in being able to identify which cells are normal self cells, which the immune system will not attack, and which cells are abnormal non-self cells, in which the immune system mounts an attack against. Now there are 3 of these genes, HLA-A, B, and C, and we each get 2 alleles of each, being diploids. So altogether we have 6 alleles. For each of these genes in the population, the human population, there's a huge number of different alleles, from 400 to 1200. So there's a huge number of possible combinations of 2 alleles for each of these 3 genes. And humans have a very low likelihood of sharing the same, exact same alleles as each other. So pretty much we're all unique with respect to these HLA antigens. All right, second topic on the list today is pleiotropy. So what is pleiotropy? First of all, a definition. It's the situation where a given genotype has an effect on more than one phenotypic trait. So one gene is somehow influencing the phenotype of more than one character. A few examples to illustrate. First example, and this is straight from your textbook, is what was discovered as a mutation found in the Aborigines in New Zealand called the Maori. Men in this population, there's a common malady in which they suffer from both respiratory problems, frequent pneumonia, for example, and are also infertile. They're sterile. So at first, these were thought to be seemingly disconnected from each other. However, they always occurred together. And what was found was that the mutation causing these defects is in a gene that encodes a part of the cilia, those tiny hairs that are found on the surface of cells lining the respiratory tract, which are important for moving mucus out of the lungs. Mutation, in which the cilia are not functional, causes settling of mucus in the lungs, and therefore they're more prone to infection. This same defect affects the formation of the flagella in the sperm. And so as a result, these men make sperm, but they're not motile. And so that's the basis for the sterility. So this is a single mutation, mutant allele, that affects two seemingly unconnected characteristics. Second example is Marfan syndrome. This is a syndrome in which there are a number of characteristic phenotypic results. For example, Marfan's individuals are very tall, and they are more likely to have defects in multiple organs. The reason for Marfan syndrome is due to a mutation in a gene called the fibrillin gene. Fibrillin encodes a protein that's important in many connective tissues. So bone and cartilage, and for example, the heart valves are made of connective tissue. People suffering from Marfan syndrome have multiple defects associated with connective tissue in general. The third example comes from mice. And in this example, there's a famous story of a mutant mouse called the yellow mouse. And I'm going to give you a little bit of background about that in a second. But first of all, this is an example of pleiotropy because it affects the fur color phenotype of the mouse, and it also affects the viability of the mouse. So it's two things, fur color and viability. It's an example of both pleiotropy and the next topic we'll consider, which is lethal alleles. So a little bit about the historical background of the yellow mouse. This was discovered in the lab of a French mouse geneticist named Lucien Quesnel. He had a colony of lab mice that have brown coat color called the agouti mouse. And then one day he found this strikingly yellow mouse, and he investigated it further. One thing he attempted to do was to make a pure breeding strain of this yellow mouse. That's a common practice of geneticists when they find a mutant strain, is to try to make a pure breeding stock that they can keep in order to study it. His attempts to do so, however, always failed, and he was puzzled about why that was. In the process of investigating the yellow mouse, he carried out two particularly important crosses, which I will go through on the subsequent slides. First cross was crossing a yellow mouse to one of his pure breeding agouti mice. And the second cross was crossing together two yellow mice. So first of all, just this slide to indicate a yellow versus an agouti mouse. And you can see the yellow mouse really stands out. The slide also includes a couple of other mutant strains for coat color, but we're going to focus in on these two. All right, here's the first cross. The first cross was between an agouti mouse and a yellow mouse. The agouti mouse was pure breeding, so he knew that mouse was homozygous. When he did this cross, he always came up with, in the progeny, a 50-50 breakdown. Half of them were yellow and half of them were agouti. He concluded a couple things from this cross and the results. First of all, that the yellow mice must be heterozygous in order to get this 50-50 result in the offspring. Okay, so he designated alleles, big A allele for agouti, and the A super Y allele indicating the yellow. All right, so the yellow mouse is heterozygous. Not only that, if it's heterozygous, that must mean that the AY allele is dominant to the agouti allele. Okay, so that was an important conclusion from this cross. Two important conclusions. With respect to fur color, the yellow allele is dominant to the agouti allele, and the yellow mouse was heterozygous. Okay, next cross. After doing the previous cross a few times, he was able to come up with both male and female yellow mice, and he crossed them together. Notice that this cross is a lot like the F1 cross that Mendel carried out in his monohybrid crosses. Crossing together two heterozygotes. When Mendel did this in the F1, he saw in the F2 a 3-to-1 ratio in the monohybrid cross. When Quinault did this, he found the mice resulting from this cross were seen in a 2-to-1 ratio, not a 3-to-1 ratio. 2-to-1 ratio in the offspring of a cross between two heterozygotes. What he found out was the reason for this unusual ratio was because of the fact that mice that were homozygous for the yellow allele were lethal. And he investigated this by finding out in the wombs of the pregnant mothers there were embryos that failed to develop beyond a certain stage. Okay, so what this means is that the yellow allele is dominant to agouti for producing the yellow phenotype. However, the yellow allele in homozygous form is lethal. So this must be a recessive lethal. It's not seen in heterozygous form. Those mice live just fine. The lethality is only seen in homozygous form. It also explains Quinault's observation or inability to obtain pure breeding yellow mice. It's because they were lethal. There's another example of this. It's pretty much the same in cats, and it's called the Manx allele. The Manx allele is dominant for producing the tailless phenotype, and on this slide there's an example of a Manx cat. This cat is heterozygous and has no tail. However, in homozygous form, it's lethal. So it's a recessive lethal. The eye phenotype here is unconnected to the tailless phenotype. Final slide here shows a table with a breakdown of the extensions of Mendelian inheritance that we've covered so far and how they compare to what Mendel saw. It's a useful table to go through and read how the observations differ from what Mendel saw. So that will conclude this video. In the next video, I will carry on with a number of other extensions from Mendelian inheritance. See ya.